Galactose is a monosaccharide sugar, which means it's a simple sugar that cannot be broken down into smaller sugar molecules. Galactose is an epimer of glucose, meaning they differ in their molecular configuration at only one carbon atom, specifically at the C4 position. The name galactose derives from the Greek word galactos, meaning milk, as it is commonly found in dairy products. Galactose is naturally found in dairy products such as milk, yogurt, and cheese. In these foods, it exists as part of lactose, which is also known as milk sugar. Galactose plays a vital role in human metabolism. It serves as an energy source through glycolysis after conversion to glucose. It's also a key component of glycoproteins and glycolipids, which are important for cell structure and function. Additionally, galactose is essential for brain development and forms part of structural molecules in cell membranes. In the next section, we will explore the detailed chemical structure of galactose. Galactose has the molecular formula C6H12O6, identical to glucose. Galactose is a hexose sugar, meaning it contains 6 carbon atoms. It's a monosaccharide, or simple sugar. The chemical structure of galactose consists of a 6-carbon ring. Let's examine its structure. The 6 carbon atoms are arranged in a ring structure, numbered from 1 to 6. Each carbon has attached hydroxyl groups, which are important for the molecule's chemical properties. The key structural difference between galactose and glucose is at the C4 position. In galactose, the hydroxyl group at C4 has a different orientation compared to glucose. In glucose, the hydroxyl group at C4 points downward, while in galactose, it points upward. This small structural difference significantly affects the properties and metabolism of galactose. This structural difference is crucial in distinguishing galactose from other monosaccharides and influences its biochemical behavior. Galactose exists in multiple isomeric forms that interconvert in solution. The open-chain form has a linear structure with a carbonyl group at carbon 1 and hydroxyl groups on the remaining carbons. In solution, galactose primarily exists in cyclic forms. The most common is the pyranose form, which has a 6-membered ring. The pyranose ring forms when the carbonyl oxygen at carbon 1 reacts with the hydroxyl group at carbon 5. Galactose can also form a 5-membered furanose ring, though this is less common in biological systems. In biological systems, these different forms have important implications. An equilibrium exists between these forms in solution, with the pyranose form predominating due to its greater stability. Different isomeric forms have different reactivity profiles, and enzymes often recognize and process specific forms selectively. Galactose, like other monosaccharides, exists in different anomeric forms that differ in the configuration of the hydroxyl group at the anomeric carbon. The anomeric carbon is the carbon atom adjacent to the oxygen atom in the ring, labeled as C1 in galactose. Galactose can exist in 2 anomeric forms, alpha-galactose and beta-galactose. The key difference is the orientation of the hydroxyl group at the anomeric carbon. In alpha-galactose, the hydroxyl group at C1 is positioned below the ring plane, while in beta-galactose, it extends above the plane. A phenomenon called mutarotation occurs when galactose is dissolved in water. This is the spontaneous interconversion between alpha and beta forms. At equilibrium in solution, about 36% of galactose exists in the alpha form, while 64% is in the beta form. The different anomeric forms of galactose have important biological implications. Enzymes often show specificity for either the alpha or beta form, meaning they can only catalyze reactions with one specific anomer. The anomeric form determines the type of glycosidic linkage that forms when galactose connects with other sugars in polysaccharides and glycoproteins. Cell surface receptors may recognize and interact differently with alpha versus beta galactose, affecting biological recognition processes. Galactose is a monosaccharide sugar that forms an important part of our diet. The primary dietary source of galactose is lactose, a disaccharide composed of glucose and galactose. Dairy products are the most significant source of galactose in the human diet, particularly milk and milk-derived products. While cow's milk contains about 4.8 grams of galactose per cup, human breast milk contains even higher concentrations, making it an essential nutrient for infant development. Galactose is also found in smaller amounts in various non-dairy food sources. Some fruits like apples, cherries, and figs contain galactose. Vegetables such as Brussels sprouts and cabbage, and legumes like kidney beans and soybeans also provide small amounts. The typical daily intake of galactose varies significantly based on age and dietary habits. Adults typically consume between 10 to 20 grams of galactose daily, while children consume slightly more at 15 to 30 grams. Infants who rely on milk as their primary food source have the highest intake, ranging from 30 to 50 grams daily. In summary, galactose is widespread in the human diet, with dairy products serving as the primary source, supplemented by various plant-based foods. Galactose metabolism is the biochemical process that converts galactose to glucose 6-phosphate. This metabolic conversion is essential because galactose cannot be directly utilized in glycolysis for energy production. Galactose metabolism primarily occurs in the liver, specifically in the cytoplasm of liver cells called hepatocytes. Once converted to glucose 6-phosphate, this molecule can then enter glycolysis, the metabolic pathway that generates ATP energy for cellular functions. The conversion of galactose to glucose 6-phosphate is necessary because galactose cannot directly enter glycolysis. This process primarily occurs in liver cells, enabling energy production from dietary galactose through a series of enzyme-catalyzed reactions. The Leloir pathway is the primary mechanism for galactose metabolism in humans. It was discovered by Luis Federico Leloir, who was awarded the Nobel Prize in Chemistry in 1970 for his work. The pathway serves a crucial role in converting galactose found in dairy products into glucose that can be used for energy production. The Leloir pathway has several important functions in human metabolism. First, it's the primary mechanism for utilizing galactose from our diet. Second, it's essential for metabolizing dairy products, which are rich in galactose. Third, it efficiently converts galactose to glucose for energy. And finally, deficiencies in this pathway lead to various forms of galactosemia. The Leloir pathway consists of several enzymatic steps. These include phosphorylation by galactokinase, conversion to UDP galactose by galactose 1-phosphate uridylyl transferase or GALT, epimerization to UDP glucose by UDP galactose 4-epimerase or GALE, and finally recycling of UDP glucose and entry of glucose into glycolysis. The key enzymes in this pathway are galactokinase, galactose 1-phosphate uridylyl transferase, commonly known as GALT, and UDP galactose 4-epimerase. Each of these enzymes catalyzes a specific step in the conversion of galactose to a form that can enter glycolysis. Understanding this pathway is critical for comprehending disorders of galactose metabolism, like galactosemia. The first step in the Leloir pathway involves the phosphorylation of galactose by the enzyme galactokinase, known as GALC. Galactokinase is an enzyme that catalyzes the transfer of a phosphate group from ATP to galactose. In this reaction, ATP serves as the phosphate donor, providing the energy needed for the phosphorylation. This reaction transfers a phosphate group from ATP to the hydroxyl group at the first carbon position of galactose, forming galactose 1-phosphate and releasing ADP. Let's examine the significance of this phosphorylation step. The phosphorylation of galactose serves several important functions. First, it traps galactose inside the cell, as the phosphorylated form cannot cross the cell membrane. Second, it activates galactose for the subsequent reactions in the Leloir pathway. Third, this phosphorylation prepares galactose for the transfer reaction with UDP glucose in the next step of the pathway. To summarize, the galactokinase reaction can be represented by this equation, where galactose and ATP react to form galactose 1-phosphate and ADP. This phosphorylation is the essential first step that begins the metabolic processing of galactose through the Leloir pathway. In step 4 of the Leloir pathway, we focus on how UDP glucose is regenerated to continue the metabolic process. The Leloir pathway forms a cyclical process where several key molecules and enzymes work together to metabolize galactose efficiently. In the previous step, Gale converted UDP galactose to UDP glucose through epimerization. Now, this UDP glucose can re-enter the GALT reaction. GALT transfers a UDP group from UDP glucose to galactose 1-phosphate. Meanwhile, new galactose enters the pathway through galactokinase, which phosphorylates it to galactose 1-phosphate, continuing the cycle. This regeneration step creates a continuous cycle where UDP glucose is constantly recycled, allowing efficient processing of galactose without constantly synthesizing new UDP glucose. This cyclical nature of the Leloir pathway is a perfect example of metabolic efficiency. The body conserves energy by recycling UDP glucose rather than creating it from scratch for each molecule of galactose processed. The regeneration of UDP glucose represents a key metabolic advantage, allowing for conservation of cellular resources while maintaining efficient galactose processing. This regeneration step completes our understanding of the core Leloir pathway cycle for galactose metabolism. In the final steps of galactose metabolism, we focus on the conversion of glucose 1-phosphate to glucose 6-phosphate. Glucose 1-phosphate, produced from the previous GALT reaction, requires further processing to become metabolically useful. The enzyme phosphoglucomutase catalyzes this critical conversion. The reaction produces glucose 6-phosphate, a key intermediate in carbohydrate metabolism. Let's examine how this enzyme works. Phosphoglucomutase transfers a phosphate group from the first carbon to the sixth carbon position of the glucose molecule. Glucose 6-phosphate is the gateway to multiple metabolic pathways. This is why the conversion from galactose is so important. Primarily, glucose 6-phosphate can enter glycolysis, where it's broken down to produce energy in the form of ATP. Alternatively, it can be used for glycogen synthesis, storing energy for later use. It can also enter the pentose phosphate pathway, which generates NADPH and pentoses for nucleotide synthesis. This final conversion completes the transformation of galactose into a form the body can readily use for energy production and various biosynthetic pathways. Galactose metabolism is tightly regulated to maintain proper sugar balance in the body. The body employs three primary mechanisms to regulate galactose metabolism: enzyme induction, feedback inhibition, and hormonal control. Enzyme induction is a key regulatory mechanism where the presence of galactose triggers increased production of enzymes involved in its metabolism. When galactose enters the cell, it activates transcription factors that bind to DNA, increasing the expression of genes encoding enzymes like galactokinase, transferase, and epimerase. Feedback inhibition is another important mechanism where end products of the pathway inhibit earlier steps in the process. As galactose is metabolized through the Leloir pathway, accumulation of downstream metabolites like glucose 1-phosphate can inhibit earlier enzymes such as galactose 1-phosphate uridylyl transferase. Hormonal control is a third major regulatory mechanism for galactose metabolism, particularly through insulin and glucagon. Insulin, released after meals, promotes galactose metabolism by increasing the activity of key enzymes and facilitating the uptake of galactose into cells. Conversely, glucagon, secreted during fasting, suppresses galactose metabolism to prioritize glucose production through gluconeogenesis. The regulation of galactose metabolism is an integrated system where these three mechanisms work together to maintain metabolic homeostasis. Enzyme induction increases pathway capacity when galactose is present in the diet. Feedback inhibition prevents metabolic overflow by slowing down the pathway when end products accumulate. Hormonal control integrates galactose metabolism with the overall metabolic state, coordinating it with glucose metabolism and energy needs. This section introduces galactosemia, a group of inherited metabolic disorders where the body cannot properly metabolize galactose. In galactosemia, patients cannot process galactose due to genetic mutations affecting specific enzymes. Galactosemia results from mutations in one of three key enzymes in the Leloir pathway: galactokinase, galactose 1-phosphate uridyl transferase, or UDP galactose 4 epimerase. These disorders vary in prevalence. Classic galactosemia, caused by GALT deficiency, occurs in approximately 1 in 30,000 to 60,000 births. Galactokinase deficiency is less common at about 1 in 100,000 births, while epimerase deficiency is the rarest form. Without proper enzyme function, galactose and its metabolites accumulate in the body, causing damage to the liver, brain, kidneys, and eyes. The severity of symptoms varies depending on which enzyme is affected and the degree of enzyme deficiency. Classic galactosemia, also known as type 1, is the most severe form of galactosemia. In normal galactose metabolism, the GALT enzyme converts galactose 1-phosphate to UDP galactose. In classic galactosemia, the GALT enzyme is deficient or non-functional. This leads to accumulation of galactose 1-phosphate, as it cannot be properly metabolized. Classic galactosemia has a clear genetic basis. The deficiency in GALT enzyme leads to several biochemical consequences. These biochemical abnormalities cause various mechanisms of tissue damage. As a result, patients with classic galactosemia present with several clinical manifestations. In summary, classic galactosemia is the most severe form of galactosemia caused by GALT enzyme deficiency, leading to toxic accumulation of galactose metabolites and multi-organ damage if not treated. Type 2 galactosemia results from a deficiency of the enzyme galactokinase, also known as GALC. Galactokinase catalyzes the first step in galactose metabolism, the phosphorylation of galactose to galactose 1-phosphate using ATP. In patients with type 2 galactosemia, this initial phosphorylation step is blocked, preventing further metabolism of galactose. Without functional galactokinase, galactose accumulates in the blood and tissues. The elevated galactose is converted to galactitol by aldose reductase. Galactitol accumulates in the lens of the eye, causing osmotic swelling. This leads to cataract formation, which is the primary clinical manifestation of type 2 galactosemia. Unlike classic galactosemia, type 2 galactosemia is generally milder. The primary clinical manifestation is cataract formation, which typically develops during childhood. The biochemical consequences include elevated blood galactose and increased galactitol production, which leads to osmotic swelling in the lens and cataract formation. Diagnosing galactosemia involves several important clinical procedures to identify the enzyme deficiencies responsible for this metabolic disorder. There are three main approaches to diagnosing galactosemia: newborn screening, enzyme activity assays, and genetic testing. Newborn screening is typically performed within the first 24 to 48 hours of life. A small blood sample is collected from the infant's heel onto a special card. The dried blood spots are then analyzed to detect elevated levels of galactose or galactose 1-phosphate, which indicate a possible galactosemia diagnosis. Newborn screening for galactosemia is mandatory in most countries due to the severity of untreated disease and the effectiveness of early intervention. A positive screening result is not a definitive diagnosis and requires follow-up confirmatory testing through enzyme assays or genetic analysis. Enzyme activity assays are the gold standard for diagnosing galactosemia. These tests directly measure the function of enzymes involved in galactose metabolism. For classic galactosemia, the GALT enzyme activity is measured using red blood cells or cultured skin fibroblasts. In affected individuals, GALT activity is severely reduced or absent. Similar assays can determine galactokinase and epimerase deficiencies, helping to distinguish between the three types of galactosemia. Genetic testing identifies specific mutations in the genes responsible for galactose metabolism. This provides a definitive molecular diagnosis of galactosemia. For classic galactosemia, the GALT gene is sequenced to identify mutations. More than 300 different mutations have been identified in this gene. Similar genetic analyses can identify mutations in the GALT1 gene for type 2 galactosemia and the GAL gene for the rare type 3 form. Early detection of galactosemia is critical for preventing serious complications. When diagnosed through newborn screening, treatment can begin before symptoms develop. Without early detection, infants may develop symptoms like jaundice, vomiting, and lethargy before diagnosis, potentially leading to permanent damage even after treatment begins. Prompt dietary intervention through galactose restriction prevents life-threatening complications and allows for normal development in many affected individuals. The primary management approach for galactosemia involves dietary restriction of galactose and lactose. We need different treatment approaches depending on the type of galactosemia. For type 1, or classic galactosemia, patients require strict, lifelong restriction of galactose and lactose. Infants need special formula without galactose, and regular monitoring for long-term complications is essential. For type 2 galactosemia, or galactokinase deficiency, patients need moderate galactose restriction with a focus on preventing cataracts. The diet is often less restrictive than for type 1. Type 3 galactosemia, or epimerase deficiency, requires variable dietary restriction based on severity. Mild cases may need minimal restriction, while severe cases should follow the type 1 protocol. Regular monitoring is crucial for patients with galactosemia. From initial diagnosis through lifelong management, patients need specific tests at different intervals. At diagnosis, initial testing establishes baseline levels and treatment begins immediately. At 3 months, galactose 1-phosphate levels are checked along with a physical assessment. At 6 months, developmental screening and dietary review are recommended. Annual monitoring includes a comprehensive metabolic panel, growth assessment, and eye examination. Long-term monitoring includes bone density testing, neurological evaluations, and fertility assessments, especially important as patients reach adolescence and adulthood. Despite dietary management, patients with galactosemia may develop various complications requiring supportive care. For cognitive complications, early intervention, special education, and speech therapy are recommended. Physical complications require growth monitoring, nutritional support, and attention to bone health. Reproductive complications, particularly in females with classic galactosemia, require hormone evaluation, fertility counseling, and specialized care. It's important to emphasize that management of galactosemia is lifelong for most forms of the condition. Dietary restriction must be maintained. Regular medical follow-ups are essential, and both patient education and family support play critical roles in successful management. A multidisciplinary care team provides the best outcomes for patients. Once converted to glucose 6-phosphate, galactose can enter glycolysis and contribute to the body's energy production. Let's trace the energy production pathway from galactose to ATP. Once galactose has been converted to glucose 6-phosphate via the Leloir pathway, it can enter glycolysis like any other glucose molecule. Pyruvate from glycolysis can then enter the TCA cycle and electron transport chain, ultimately producing ATP, the cell's energy currency. Let's analyze how much ATP is produced from a single galactose molecule. While converting galactose to glucose 6-phosphate requires energy, no net ATP is produced in this preparatory phase. During glycolysis, glucose 6-phosphate is broken down to pyruvate, generating 2 ATP through substrate-level phosphorylation. The conversion of pyruvate to acetyl-CoA doesn't directly produce ATP but prepares molecules for the TCA cycle. The TCA cycle produces 2 more ATP through substrate-level phosphorylation, along with reduced electron carriers. The electron transport chain generates the most ATP, approximately 28 per galactose molecule, through oxidative phosphorylation. In total, complete oxidation of 1 galactose molecule yields approximately 32 ATP molecules. How does galactose compare to other dietary sugars in terms of energy production? Galactose and glucose both yield 32 ATP molecules when fully metabolized. Fructose yields slightly less at about 30 ATP due to differences in its initial processing. Lactose, a disaccharide composed of galactose and glucose, yields around 60 ATP when both monosaccharides are fully metabolized. It's important to note that galactose must first be converted to glucose 6-phosphate before it can generate energy. This conversion process is less direct than glucose metabolism. Despite this additional conversion step, galactose ultimately provides the same energy yield as glucose. making it an important energy source, especially from dairy products. Galactose serves as an essential building block in the synthesis of numerous complex biomolecules. These include glycolipids, glycoproteins, and proteoglycans, each with distinct structural and functional roles. Glycolipids are lipids with attached carbohydrate groups, often containing galactose. These molecules play crucial roles in cell recognition, membrane stability, and signal transduction pathways. Glycoproteins are proteins with attached oligosaccharide chains that frequently contain galactose residues. These molecules are critical for cellular recognition, immune function, hormone activity, and blood group determination. Proteoglycans consist of a protein core with attached glycosaminoglycan chains that contain alternating sugar units, including galactose. These large, complex molecules provide structural support in cartilage, enable water retention in tissues, facilitate cell migration, and bind growth factors. Galactose-containing biomolecules serve critical functions throughout the body. They enable cell-to-cell recognition and adhesion, which is essential for tissue formation and immune response. They provide structural support and maintain tissue integrity, particularly in cartilage and connective tissues. They facilitate cellular signaling and communication, allowing cells to respond to their environment and coordinate activities. Galactose plays crucial roles in development, particularly for infants and young children. Galactose is essential for brain development, particularly in the formation of myelin sheaths and cerebrosides. These galactose-containing compounds are critical for proper myelination, which ensures efficient neural signaling and supports neuronal connectivity. Galactose is a key component of glycoproteins and glycolipids on cell surfaces. These galactose-containing structures are crucial for cell-cell recognition, adhesion, and communication, which are essential for proper tissue formation and organization during development. Galactose also plays a significant role in immune system development. Galactose-containing glycans are crucial for pathogen recognition, antibody-antigen interactions, and the development of proper immune tolerance during early life. The importance of galactose is most pronounced during early development, particularly in the first 2 years of life. This represents a critical window when brain growth is most rapid and galactose demand is highest. Galactose metabolism deficiencies are most harmful during this critical developmental period, which is why early diagnosis and management of galactosemia is crucial. Galactose has several emerging therapeutic applications beyond its primary role in metabolism. One important application is in treating certain metabolic disorders. UDP galactose supplementation has shown promise in conditions like PGM1-CDG, where it helps enhance glycoprotein synthesis. Galactose plays a significant role in diagnostic imaging applications. Galactose-based contrast agents are used to assess liver function and for receptor-targeted imaging techniques. Researchers are exploring galactose's potential in treating specific diseases. It shows promise in cancer therapy, targeted drug delivery systems, and potentially in neurodegenerative disorders. Future research in galactose therapeutics is focusing on glycoengineering approaches, personalized therapy, and novel delivery mechanisms to expand its clinical applications. These diverse therapeutic applications highlight galactose's potential beyond its traditional metabolic role, offering promising avenues for medical treatments and diagnostics. Research in galactose metabolism continues to advance in several exciting directions. 3 key areas that show particular promise include gene therapy for galactosemia, new regulatory mechanisms, and applications in precision medicine. Gene therapy represents a promising approach for treating galactosemia at its genetic roots. CRISPR-Cas9 and other gene editing technologies are being investigated to correct mutations in genes responsible for galactose metabolism disorders. Current research focuses on 3 primary target genes: GALT, responsible for classical galactosemia, GALK1 for galactokinase deficiency, and GAY for epimerase deficiency. Beyond gene therapy, researchers are uncovering new regulatory mechanisms that control galactose metabolism. Recent discoveries have identified complex cellular processes that regulate how galactose is metabolized within cells. These include transcriptional regulation, microRNA control mechanisms, post-translational modifications of key enzymes, and nutrient-responsive signaling pathways. Perhaps most exciting is the emerging role of galactose metabolism in precision medicine. By analyzing an individual's genetic profile and galactose biomarkers, medical treatments can be personalized for optimal efficacy. This approach shows promise for early disease detection, individualized treatment plans, therapeutic monitoring, and novel drug development for galactose-related disorders. Looking ahead, research in galactose metabolism is expected to follow an ambitious trajectory. From our current understanding of basic mechanisms and clinical treatments, the field will move toward improved gene therapies, advanced regulatory maps, and validated biomarkers within the next 5 years. The next decade may bring personalized treatments, pathway engineering, and preventative approaches, ultimately working toward a complete cure for galactosemia and novel therapeutic applications. In this summary of galactose metabolism, we'll review the key aspects of this essential pathway and its significance in human health. The Leloir pathway is the primary route for galactose metabolism, converting galactose to glucose for use in energy production. The pathway involves 3 critical enzymes: galactokinase, galactose 1-phosphate uridyl-transferase, and UDP galactose 4-epimerase. Defects in the Leloir pathway lead to galactosemia, a group of metabolic disorders with varying clinical presentations. Classic galactosemia, or type 1, results from GALT deficiency and causes the most severe symptoms, including liver damage and developmental delays. Type 2 galactosemia, caused by GALT deficiency, primarily leads to cataract formation and has a milder presentation. Type 3 galactosemia from GALT deficiency has variable symptoms depending on the level of residual enzyme activity. Galactose metabolism serves multiple critical biological functions. In energy production, galactose is converted to glucose, which can then enter glycolysis to generate ATP. Galactose is also essential for the synthesis of complex biomolecules, including glycoproteins, glycolipids, and proteoglycans. Furthermore, proper galactose metabolism is critical for normal brain development and neurological function. To fully understand galactose metabolism, we must view it as an integrated system connecting biochemistry, clinical medicine, and physiology. The Leloir pathway provides the biochemical foundation with its 3 key enzymes orchestrating the conversion of galactose. Defects in this pathway lead to various forms of galactosemia, with clinical manifestations ranging from mild to severe. The pathway serves critical biological functions in energy production, biomolecule synthesis, and development. Understanding regulation and treatment options, particularly dietary management, is essential for clinical care. In conclusion, galactose metabolism represents a perfect example of how biochemical pathways directly impact human health and disease, connecting molecular mechanisms to clinical outcomes.